Hi there, and welcome to part 2 of molecular expression or gene expression. So we've talked about, so far in genetics, we have talked about, of course, structure and also replication. And so what I want to introduce to you today is the stages of transcription. We're going to talk about how we go from DNA to mRNA to messenger RNA so that it can later then be translated, which is part 3, into a protein. And we're also going to talk a little bit about we're going to compare prokaryotic and eukaryotic transcription. We're also going to look at what's known as RNA processing and introduce in eukaryotic cells some additional steps that occur in transcription that do not occur in prokaryotes. So we're going to initially look at prokaryotic transcription. We're going to look at the 3 stages, which are the same 3 stages that occur in eukaryotic, just the mechanisms differ a little bit. So we're first going to study how transcription occurs in a prokaryotic organism and then apply those principles to eukaryotic transcription. So transcription occurs in 3 stages: initiation, elongation, and termination. And we'll see these same 3 stages when we look at translation. Now in initiation, this is stage 1. And in initiation, this is also referred to as a recognition step. Now I want to point to this piece of DNA up here, and I want to show you a couple things. So in pink, we have the DNA strand that is being transcribed. In green, we have the actual gene itself. And in blue, right here, we have a region known as the promoter region. And the promoter region is a sequence of DNA that is going to be recognized by a transcription factor. Now, you may remember the term transcription factor from Unit 2. When we studied cell signaling, the whole point of all of those long signaling pathways that we looked at, remember the RTK pathway and the EGFR pathway, when we looked at all of those different pathways, one of the ultimate goals of it was the cell response. And that cell response was initiated because of our cell signaling pathway activated what was referred to as a transcription factor. So the transcription factor is a protein that's going to recognize the promoter sequence of a specific gene. So transcription factors, there's a multitude of them. We have thousands of them in our cells, and they are proteins that are responsible for recognizing a unique sequence or a specific promoter sequence of a gene, a specific gene. So different transcription factors will be used to regulate different genes. Just beyond the promoter sequence at the edge of it, once our transcription factor kind of recruits everyone and says, hey, we need to get this started over here, in bacteria, another protein known as the sigma factor. The sigma factor is required, will bind to the transcription factor, and will activate RNA polymerase. So RNA polymerase is our final enzyme to attach to this sigma factor and transcription factor protein. All of these proteins are all kind of congregating here, right at the edge of that promoter sequence, in order for RNA polymerase, which is this large yellow bubble here, for RNA polymerase to be activated and begin the transcription process. RNA polymerase, by the way, structurally is very, very similar to DNA polymerase. Now in elongation, once the RNA polymerase gets activated, so that transcription factor bound, the sigma factor bound, and RNA polymerase is now attached and activated. It's ready to start. Its transcription process. The sigma factor will fall off. When sigma factor falls off, that signals the end of initiation and the beginning of elongation. This allows now our RNA polymerase to move forward and create what's called an open complex. So remember, the pink DNA strand is what we are actually transcribing. The green strand is the actual gene itself, and we will look at why we have to transcribe the bottom in a second. So this transcription, what's sometimes referred to as a transcription bubble or open complex, is where RNA polymerase is separating the 2 DNA strands. It's breaking the hydrogen bonds between the 2 DNA strands in order to access the nucleotides in the center. It must base pair RNA nucleotides to the DNA nucleotides so that the mRNA transcript has the same nucleotide sequence as the actual gene. So that's creating the open complex, and it does so through base pairing. But when it base pairs with what's referred to as the template or coding strand. And the template or coding strand is the complementary strand to the actual gene itself. So the green strand up here is the actual gene sequence. But if we tried to transcribe directly on the gene, we would actually get the complement of it. So we have to take the mirror image of the mirror image. And let me show you what I mean up here. So here I have a DNA sequence. If this is my 3 to 5 prime, then my 5 to 3 prime would be this, A, T, G, C, C, T. Now, if I am a RNA polymerase, if this is the gene right here. Now, remember, the gene is only located on one strand. The gene is not both strands. It is only the sequence found on one strand. And if the bottom, the 5 to 3 prime strand is the actual gene sequence itself, then as RNA polymerase, I need to transcribe the opposite because I want to replicate the gene. I want an RNA transcript that's going to represent my gene, right? But I can't do that if I'm complementary base pairing directly to the gene. I can only do it if I complementary base pair to the complement of the gene. So let me show you what I mean. If I were to as RNA polymerase, if I were to complementary base pair to the gene itself, then the actual sequence I get is the complement. This, in RNA language, matches that, and that's not the gene. This is the gene, right? And I need that gene, so I'm going to complement to the what we call the coding strand. This, in DNA language, is now the same as my gene. U always replaces T. So I just copy my gene and I replace all the T's with U's. But in order to do that, this is done through base pairing. So I have to base pair the mirror image of the mirror image. So that's what's meant by this template or coding strand. The non-coding strand is not used because the non-coding strand is the actual gene itself, or the gene sequence. Our mRNA transcripts are always synthesized in the 5 to 3 prime direction. Uracil is always substituted for thymine, and behind that open complex that's created by the RNA polymerase, that open complex, the DNA is going to zip right back up because these bases are still intact and complementary. So that's going to cause that DNA molecule to zip right back up behind RNA polymerase. So that only about 10 to 15 base pairs are opened up at a time. And when they are in that open complex, it is within the actual RNA polymerase enzyme itself. So in stage 3 of transcription, this is the termination sequence. And in termination, RNA polymerase will actually run into a sequence that signals termination of the actual gene itself. And what will happen is that RNA sequence will create this stem loop-like structure like this, and there's a helicase that kind of reads along it, and it causes the RNA polymerase itself to slow down and kind of wobble a little bit and then eventually fall off of the actual DNA molecule. So there will be some bases over here that get transcribed, but they get later cut out by that stem loop type structure. So here we have initiation. Here's where that open complex is formed. Notice that we still have sigma factor bound to RNA polymerase getting it started. Once the sigma factor falls off, that means that RNA polymerase is stabilized, the open complex is ready, and we now start our base pairing. So here we have our direction from 5' to 3', and you can see that it is complementary base pairing each RNA nucleotide to its complement DNA nucleotide. And this, of course, creates this big, long mRNA transcript. Once it reaches that termination sequence, then termination will occur. The RNA polymerase will dissociate or come off of the DNA molecule, as well as release the completed mRNA transcript so that the transcript can then later be processed. Now, this is here we're looking at prokaryotic transcription, and I just want to let you know, in prokaryotic transcription, while the mRNA transcript is being released, because prokaryotes do not spatially separate the transcription process from translation or protein synthesis, they will begin the protein synthesis process at the same time that the mRNA transcript is being made. So for prokaryotes, transcription and translation are simultaneous.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so now I want to talk a little bit about the direction of transcription and how the DNA strands are used in transcription. Now, DNA transcription is occurring on one strand of the DNA. Even though DNA is double-stranded, the actual gene sequence itself is only located on one strand. And in all cases, RNA transcripts are going to be transcribed, they're going to be synthesized from 5 to 3, which means the DNA template, the strand that we're reading, the coding strand, is always the 3 to 5. Now, here we have a large DNA molecule, right? We have, say, a region of a chromosome. We have 3 different genes that are all located on the same chromosome. And if we look at the bottom here, the top strand, if we flatten out our DNA molecule so we can look at it up close, the top strand is the 5' to 3' strand in all instances here because they're all on the same chromosome. And for gene A in blue is the promoter sequence for gene A and gene B. So gene A, the promoter sequence is on the left. And that is because in this case, the gene is located on the bottom strand. And the bottom strand is the 3 to 5. And of course, the template strand is always read 3 to 5. So in this case, transcription is occurring from left to right. So that bottom strand is our template here, is our actual template, so that the top is our gene. So this is going to give us that 5' to 3' gene A RNA, right? So now we have our mRNA transcript for that gene. If we go over to B, B is the exact same case. Our promoter here is on the left. We know that our bottom strand is the 3 to 5, and this is the one that's going to be read. So transcription is going to occur from left to right. And that this top strand here is our gene. Now, if we move over to our third gene here, gene C, here our promoter sequence is on the right. And the reason for this is that our gene is actually now on the 3 to 5 strand, so we have to transcribe the 5 to 3. But because we can only read the DNA molecule from 3 to 5, instead of moving left to right, it's now moving right to left. So that the top strand now becomes the template strand. That's the strand that we're going to be reading because the gene sequence itself is the bottom strand. So in this case, we still end up with that 5' to 3' sequence, but we're doing it off the opposite strand. Okay.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription. resulting in the full-length mRNA transcribed from the gene.
So in eukaryotic transcription, that was all prokaryotic transcription. So in eukaryotic transcription, the features are identical. We have initiation, elongation, termination. We have a promoter sequence. We have the genes. We have coding and non-coding strands. We have termination sequences. All of that is the same. However, we use many more proteins to carry out those steps than are used by prokaryotes. Now, first of all, we have 3 different forms of RNA polymerase. Now, RNA polymerase 2 is responsible for transcribing mRNA, but RNA polymerases 1 and 3 transcribe genes that are referred to as nonstructural genes, and these are genes that encode for ribosomal RNA and transfer RNA. Ribosomal RNA and transfer RNA are both going to be used during translation. RNA polymerase 2, that transcribes our messenger RNA, is going to require 5 general transcription factors for initiation. So whereas the prokaryotic cells really only required 1 transcription factor, and they really just require that sigma factor, in eukaryotic cells we have 5 general transcription factors, which is the equivalent of kind of 5 sigma factors, in addition to transcription factors that are specific to specific genes. We also must process our RNA. RNA does not come directly off of the DNA and go directly into translation or protein synthesis. Our RNA must be processed, and it goes through 3 different processing steps. These are known as splicing, 5' cap, and poly A tail, and we'll look at each of those. Now, our RNA transcripts are made out of 2 different types of regions. They're all RNA nucleotides. There's no differences in the nucleotides, but different regions of our mRNA transcripts. Some of those areas are called introns, and some of those areas are called exons. And introns are regions of DNA that get transcribed off of the original DNA molecule into RNA, but are never going to be translated. They're not going to be taken out into the cytoplasm to become part of a protein. Exons, on the other hand, are the actual coding sequences that are found in the final mRNA transcript after processing. Now, when we go through splicing, the first step here, we remove the introns and bring the exons together to create one single mRNA transcript. And then, of course, we'll also have the other modifications that we will look at. So here we have a eukaryotic gene. Here's our promoter sequence over here in terminator. But in the middle is everything that's going to be transcribed. So when transcription occurs, we have a really long mRNA molecule. The first thing that's going to happen is at the 5' end, it'll be capped. And we'll talk about that process in a few minutes. But then we're going to go through the splicing process. And in the splicing process, these 2 green regions called introns are going to be removed. And they're going to be removed by a set of proteins that, when they come together, are referred to as a spliceosome. And the spliceosome is made out of multiple proteins, I believe it's 4 or 5, called snurps. That's what they're referred to as. They're referred to as snurps. And once these introns are cut out by the spliceosome, then we'll add a poly A tail, and those exons are brought together. And now you can see we have a much shorter mRNA transcript that has its 5' cap, its poly A tail, and it does not have any introns. The introns have been removed. So here's the process of splicing. And what's happening here is this intron in green is being pulled out, and the 2 exons are going to be pulled together. So they're kind of showing you the pulling together of it. And this loop structure right here is called a lariat. The lariat is formed when the spliceosome proteins come together and pull these 2 regions right here and right here. You're going to pull those together. These are known as the 5' splice site and the 3' splice site. And they are RNA sequences that are recognized by the spliceosome. So let's take a look at a spliceosome. So we have lots of introns found in our genes. Prokaryotic cells do not have introns. They're extremely rare. Whereas in eukaryotic genes, we have lots of them. So here we have an exon in yellow, another exon over here in yellow, but this green area in the middle is an intron. And in the middle of the intron is a sequence known as a branch site. That branch site is going to be recognized by the first snRNP, and that's what this large blue protein is here. And this large snRNP will recognize that branch site and start recruiting other proteins, other snRNP proteins that recognize the 3' and 5' splice sites. And when they get recruited, they pull them together. So you can see how we're now starting to get this loop of the intron there, and we've got two snRNPs attached to the 5' and 3' splice site, respectively. Eventually, these snRNPs are going to come together. When they bind to each other, it causes a conformational change that, in hanging on to that intron, pulls that intron into our now lariat structure. So it creates this lariat, and down here at the bottom, these snRNPs here at the splice sites will cut at the splice site and bring together, glue together, well, ligate would be a better word, the two exons that were on either side of the intron. And then that intron is removed, broken down, and the RNA nucleotides will be recycled. So the introns are removed from that pre-mRNA by the spliceosome, and the spliceosome again is made up of these nuclear RNA set of proteins known as snRNPs. So the intron RNA is defined by those sequences. And remember, right in between an intron and exon will be a splice site. And that splice site, when we talk about sites in DNA or in RNA, we're talking about very unique sequences. The spliceosome subunit proteins, those snRNPs, recognize those sequences. And so they'll pull those sequences, they'll attach to those sequences and then start to congregate together so that any RNA that's found between them will be pulled up in that lariat-like structure. So the spliceosome subunits recognize that. And the binding causes that loop or that lariat formation. The two exons get brought together and eventually spliced together. Now in eukaryotic transcription, when we get into RNA processing, we do have a process known as alternative splicing. And in alternative splicing, the spliceosome will be regulated. So which sequences are recognized as splice sites could be altered so that a single gene, through splicing, alternative splicing, a single gene could be spliced differently multiple times to encode for 2 or more different polypeptides. Now those polypeptides are usually of very similar function. They don't really vary a whole lot in function, but they are different polypeptides. So the introns that are found in ribosomal RNA and in transfer RNA are what are referred to as self-splicing. They don't use the spliceosome. Spliceosome is strictly for messenger RNA. And finally, we have 2 more processes that occur. We have what's called the 5' cap. The 5' cap is a 7-methylguanosine. You can see here, they're showing it to you here in this green here. This 7-methylguanosine is going to be covalently attached to the 5' carbon at the 5' end of a mRNA transcript. This 5' cap is necessary for 2 things. One, it is required for the mRNA molecule to be able to leave the nucleus. So guard cells around those nuclear pores will not allow an mRNA transcript to leave the nucleus unless it has a 5' cap. The second function of the cap is for ribosomal recognition. A ribosome needs a 5' cap in order to actually bind to and recognize the messenger RNA transcript. So 5' caps are extremely important. The second is the 3' poly A tail. So at the opposite end of our transcript, about 100 to 200 adenine nucleotides are going to be just kind of added on. And they call it a poly A tail because we have this big long tail of adenine nucleotides at the 3' end. The whole point of the poly A tail is to provide stability and lifespan of the mRNA molecule. We don't want our mRNA molecule during translation to start getting eroded, and that can happen just like it does in replication. So as we added telomeres to the ends of DNA molecules during replication, in transcription we add poly-A tails, and the poly-A tail gives stability and increases the lifespan of the mRNA transcript when it's in the cytoplasm, out in the cytosol. Neither the 5' cap or the poly-A tail are part of the gene sequence. Those are added after transcription. They are not a part of transcription. So that's the lecture on transcription, part 2. In part 3, we're going to go into translation and how protein synthesis occurs. But for this particular one in transcription, I want you to kind of reflect on some of these questions to help you kind of review the material. What are the 3 stages of transcription? How are base pairing rules in transcription similar to the base pairing rules used in replication? How is RNA polymerase similar to DNA polymerase, and how is it different? How do prokaryotic and eukaryotic cells differ in their transcription? What is RNA splicing and what is a spliceosome? What's the difference between an intron and an exon? And what are the structures needed on a finalized mRNA transcript in order for it to leave the nucleus? So these are all just kind of a little bit of a review or recap on what was covered in this lecture. All right, guys, I'll see you on the flip side in part 3.